Yeah, good morning, everyone. Yeah, who all are present there in the class? Good morning. Is my screen visible and voice audible to all of you? If it is audible, then we can start up with the class. Okay. Yeah, good morning. Seems to be a Russian surname. Russian, this thing. Okay. So, since now we have finished and covered the chemistry and metabolism of carbohydrates. Yesterday, I finished up with the discussion of regulation of blood glucose. So, those students who are joining for the first time, if they want to go through the metabolism and chemistry of carbohydrates, they can switch over to the older videos. Today, we are going to start up with a very interesting topic: introduction to proteins and amino acids. Right? So now see, introduction to proteins and amino acids. Now see beta, first of all, what is the biomedical importance of proteins? Why we eat proteins? Because carbohydrates, I told you, carbohydrates are the universal sources of energy. For example, glucose is there, galactose is there, fructose is there. What are these proteins? And actually, proteins are simply the polymers of the amino acids. Proteins are polymers of amino acids. Okay, so basically, we have to focus on what are these amino acids, what are their uses, how they form a protein, and then we come over to what is the significance. And the most important thing about proteins is, because normally we have carbohydrates, we have proteins, we have fats in the diet. Proteins are considered to be having, proteins have maximum SDA, specific dynamic action. Very important. Action. Right. Then what are, if we talk about biochemistry, we all know that biochemistry is the study of the biochemical reactions, and you will be surprised to know that all the enzymes are basically proteins, except yes, you can say ribozymes are there, but apart from this, all enzymes are proteins. So if we talk about the biomedical importance, I can say enzymes except ribozymes are proteins. Right? Then if I talk about the antibodies which fight against the antigen, normally क्या होता है कि when a foreign particle, you can say, invades our body, there are certain defensive mechanisms present in our body, which are the antibodies. So antibodies are also proteins, or we can call them as immunoglobulins. They are also proteins. Right. Similarly, the most important function of proteins is they help in transportation. So, transport proteins are there, right? Jesse albumin is there, and you will see wherever I am talking about any carrier molecule, that carrier molecule is nothing but the protein. Okay. Then, apart from this, plasma proteins are there. The most important one, flavoprotein, hemoglobin, right? We know hemoglobin is what? It is the respiratory pigment. It is responsible for the transport of oxygen from head to toe. So you can just imagine that what is the significance of this. And that is why we have proteins in a diet also. And you know, proteins are also synthesized in the body. Right. So basically proteins are not synthesized beta. Proteins are the ones which are formed from amino acids, and these amino acids are going to form the proteins. So whatever characteristic, whatever property this protein will be having, that will come from what? Amino acids. So here we have to target only the amino acids. Clear? So if I talk about if I talk about what are these amino acids, right? अब these are a very interesting, I should say, the metabolites, because हम लोग what are we are having in our diet? We are having the proteins which are degraded to amino acids which undergo absorption. मतलब बिल्कुल it is almost parallel to what we studied in carbohydrates. Carbohydrates also we studied the same thing, no? Polysaccharides are broken to monosaccharides and these monosaccharides undergo absorption. So same thing we are going to study here. How do we define these? Amino acids are the molecules. Okay, are the molecules which have, or which can behave as an acid and a base. Right? And if I write the generalized structure of an amino acid, it is like this. Or we can say this is your alpha carbon atom. So we can say amino acids are the ones which have a alpha carbon atom where both the functional groups are attached. If we talk about, this is the amino group and this is the carboxylic group. And on the basis of this are different classification of these amino acids are there. So we can also call them as alpha carbon molecules. Right. What is an acid? We all know that acid is the one which can donate a proton. And the base is the one which can accept a proton. Right. And if you look at the structure of the amino acids, they are capable to do both things. Means on the one hand, if they are donating a proton, on the other hand, they are accepting a proton. And these type of molecules which can donate a proton or accept a proton are known as amphoteric substances. Right? So what are amphoteric substances? Amphoteric substances are the ones which can behave as an acid as well as a base. Is it clear? Right? And this is very important. And why these proteins or amino acids are basically better important, because nowadays if we talk about gene sequencing, gene expression, gene structure has to be seen. And how it is seen? Only when we consider the structure of the amino acids. Clear? Let me see who all are present here today in the class. Vrushali, are you there? Vrushali, then all the students who've been attending my classes for the past 1 week. Okay, right. Okay. So, ready? Have you joined the class today? Because for FMGE students also, this is a very important topic. In fact, for FMG students, for NEET PG students also, these amino acids are very important and questions have been asked on this. Right? So, They are the amphoteric substances. Now see, there is a term called zwitterion here. Right? What is this zwitterion? This is the ionic form. NEET UG student. Okay, good morning, Asif. Yes, for NEET UG students also it is very helpful because you also study, the last chapter in your books, applications in daily life. So this is also very important. So I will be focusing, in fact, on all the disciplines. Right? So if we talk about zwitterion, this is the structure. COO negative means they are existing in a ionic form. Clear? So now see, what happens is whenever these amino acids are allowed to run, because you know how we separate, just say most of you are fresh pass outs, you have just cleared class 12th. Yeah, good morning, Jessica. I think Jessica, you are also attending the class for the first time. Right. So what I'm trying to tell you here is that means what happens is since these amino acids, right, they are having, yeah, hi, good morning, Dilkush, how are you doing? I hope सबको सब कुछ समझ आ रहा है. This is very important. समझ आ रहा है बेटा दिलकुश? Okay, Jessica. Right. So, Jessica, till now we have finished, covered up the metabolism of carbohydrates, which you can see later. And this is, we have started up with a new topic. And yeah, good morning, Shreya. Right. So, zwitterion is simply, you can say, that whenever we try to separate the amino acids. Okay. Thank you so much, Dilkush. Crystal clear. Good. Right. So, when we try to separate these amino acids, on the basis of electrophoresis means when charge is there, because obviously you know positive charge, negative charge is there, and when they are separated by electrophoresis, we all know that they will move towards the cations, cathode, or the anode. Okay, we will allow them to run. Up, if we talk about the amino acids, this is the structure which is present in all the amino acids. Only R group is different. Okay, so depending upon the R group, their movement will vary. Getting my point? That amino acids are having zwitterion structure, a negative charge and a positive charge, and they are made to run in electrophoresis. So they will move towards the cathode, they will move towards the anode. So depending upon the R group, they are going to move. Right? But a stage will come when there will not be any migration. Why? Because the positive charge will be equivalent to negative charge, right? And when this happens, when the positive charge is equivalent to the negative charge, the amino acid will be separated. And that point is known as isoelectric point. Very, very important. Right? So I can simply define this isoelectric point as the point at which no net migration is there. No net migration of molecules is there. Oh, thank you so much. This is the best compliment I can ever have. Okay, thank you so much for the compliment. I think you are attending the class for the first time, if I'm not wrong. Ayor, I'll call you Ayor. So, best is attend the classes on a daily basis. Then only you will be able to understand. Because obviously, you people have an access to YouTube. You can watch it after that also. But my sincere advice to you is when the teacher is sitting in front of you, if you have any queries in your mind, you can ask there and then. And regarding this compliment, I am going to frame it and I am going to literally mount it on my wall. Thank you so much. Right. So coming over to this isoelectric point, the point at which no net migration of molecules is there. That means we can say that the positive charge is equivalent to the negative charge. Right. Or we can simply say the molecule is neutral. So all amino acids will be always separated at their isoelectric point. This is one very important thing which you have to understand. Because now why it is important? It is important just because of one simple reason, that amino acids are the ones which are forming the proteins. If we are talking about any disease, for example, just now I told you all the enzymes are also proteins except ribozymes. So if an enzyme is not being expressed in a particular person, we can go to the sequence of that protein or the sequence of the amino acids. So how we will come to know what is the sequence? Means on first position what is present, on second position, third or fourth. This is made possible only by the separation techniques. These separation techniques are electrophoresis is there, chromatography is there. So what is that property of that particular amino acid? The property is isoelectric point. Iso means same. Electric, basically it has come from the electric current. And point is when they are separated. Right? So this is what you have to understand. And if I talk about, say, this is your, I'll simply write the structure of the amino acid. This will be your zwitterion, right? And if it is acidic, in the acidic medium, it will be protonated, right? And here it will be in the alkaline medium, it will be deprotonated. Right? Here COO negative will be predominant and here NH3 positive will be predominant like this. H, and here it will be CNH2, COO negative, H and R. Right? So in the alkaline pH and in the acidic pH, and this is at neutral pH, they will act like this. I hope this is clear. Yes, Asif, R group is going to affect isoelectric pH. Why? Because if we talk about the amino acids, I'll just tell you after this, there are 20 standard amino acids. The difference, what is the difference? The difference is basically in the R group only. Yes, you are right. You can follow all the notes which are there in my Telegram channel and you will clear your exam with good marks. That much surety I can give. So Asif beta, R group is different. Otherwise, if we see, all the 20 standard amino acids will be the same. The only difference will be in the R group. Slowly, when we will discuss the metabolism, I will be discussing the structures also of amino acids and I'll show you only the R group will vary. Got my point? So yes, to your answer question, R group is going to affect isoelectric point. In fact, isoelectric point is based on R group. If a amino acid is bulky, for example, bulky समझते हो, so many groups are attached, it will move slowly. If it has only one group attached to it, it will move fast. जैसे if we compare a running status of a fat person and a lean person, a fat person will come around, say, he will just move slowly, whereas a fat and a lean person will immediately run. Okay, so again, R group, if it is bulky, जैसे lysine is there, arginine will they will take ages to reach. On the other hand, if glycine and alanine is there, they will reach fastly. Okay, so yes, R group will affect the isoelectric point. So this is very important. When it is neutral, it is existing as zwitterion. I'll write it here also. Very important. But when it is acidic, when pH is less than 5, less than 7, then it is acidic, and when pH is more than 7, this is alkaline. So the movement will be like this. Right? Now, after isoelectric point, then I will come over to the classification of amino acids. Then your query will be completely clear. Now, always remember, beta, there are 20 standard amino acids. Okay. Now, what do we mean by the term 20 standard amino acids? Standard means which are already existing. Okay. Now, 20 means matlab they are the only ones from where different other amino acids are derived. So I can say that from these 20 standard, there are derived amino acids also. There are non-standard amino acids also, depending upon their functions. Right? But after, the discovery, because, scientists always keep on discovering some things. So they also discovered 2 more amino acids, which added into this 21st amino acid, right? And 22nd amino acid. Okay. Now you will simply think, obviously you will think that why madam has written only 20 standard amino acids, why she is not saying 22 amino acids, isn't it? So 21st is separate, 22nd is separate. Now why it is separate? Because they are coded by nonsense codons. Very important. 21st amino acid is known as selenocysteine, right? And 22nd amino acid is known as pyrolysine. This is coded by UGA, which is obviously a nonsense codon. And this is coded by UAG, which is also a nonsense codon. Right? So I can say that there are 2 more amino acids which are added to the list. 21st, which is selenocysteine, and 22nd, which is pyrolysine. Now the question again, what is the significance of selenocysteine? Isn't it? Why we have to put it under amino acids? So the answer to this is this selenocysteine. Now always keep one thing in mind wherever we write selene. Seleno comes from selenium. So it is basically selenium plus cysteine. Right. Selenium is a very important trace mineral which is used as an antioxidant. Okay. Selenium is the trace mineral which is used as an antioxidant. And cysteine also, cysteine we will discuss, it is a sulfur containing amino acid. So it has SH group. So it is also acting as a good reducing agent. So obviously there will be importance of this. And this selenocysteine is synthesized in the body. Right? And ATP is required. And what is the significance of this? What is the importance of this? Selenocysteine is a component of, right? It is a component of so many important enzymes in the body. For example, you have to just learn the name of the enzymes better, because this is for first year students especially and for UG students especially. This is a new concept. Okay, but slowly if you study, if you go through the entire lectures, then automatically the things will be clear to you, so you don't have to worry about that part. And yes, you have to mug up certain things, then only it is made possible to remember. Okay, so selenocysteine is a component of this enzyme, thioredoxin reductase, right? This is a very important one. Then deiodinase, and in the metabolism of glycine. Clear? So these are the important features of this. Glycine reductase, chalo, I'll write the name of the enzyme here. Okay, so selenocysteine is important. So if somebody asks you, if you get a question in the exam like this, that how many type of amino acids are there? So you have to write there are 20 standard amino acids. Along with 20 standard amino acid, 21st is there, which is coded by UGA, and its name is selenocysteine, and it plays a very important role in the body by acting as a coenzyme. And 22nd is pyrrolysine, which is also coded by a nonsense codon. So overall, there are 20 standard plus 21st and 22nd amino acids. Clear? And this is a very important question which is asked in the exam, that what is isoelectric point? So what is isoelectric point? Iso means same, when the charges are the same. Same means neutral. When the amount of negative charge is equivalent to amount of positive charge, we will simply say that it is basically separated on that isoelectric point. Right, clear? Now after this, then we come over to the classification. Now there are different ways of classifying the amino acids. The first way is as per R group. Asif, you were talking about this classification, this we will discuss now. So as per R group. Second is as per nutritional requirement. Right? And obviously as per the charges. Right? And the last one is as per their, this is very important, their metabolic fate. Very important. Right? Now, first of all, we will take these all we are going to take up in detail, because these are very, very important again. So if we talk about, first is as per R group. Right? The first way of classification is R group. Now see, this is the structure. I'll just write NH2 and COOH, R. Okay. Now this R group can be aliphatic, All of you know what are aliphatic compounds. Aliphatic are the straight chain compounds. Okay. So aliphatic will include glycine, right? Alanine, leucine, isoleucine, and valine. So they will come under aliphatic amino acids, which are straight chain amino acids. Here. Yes, you can say Gavali, you can, yes, very good. You can make it, remember it as Gavali. Mnemonics, beta, you can make on your own also. Okay, mnemonics basically, if you try to learn, memorize biochemistry in the form of mnemonics, it will be very easy for you people to understand. So you can make mnemonic like that. So Gavali is glycine, alanine, valine, leucine, isoleucine. And here, this villi, they are branched chain amino acids also. Okay, because the main concept here to understand is the metabolism, the most important one. And that is why their structures are made. So, coming over to this ASIF, glycine will have its own isoelectric pH, alanine will have its own, leucine will have its own, but every amino acid will have its own isoelectric pH, or उसको हम बोलते हैं PI. Then after aliphatic, then we come over to aromatic. Aromatic means obviously they have an aroma or they have a benzene ring attached to them. So aromatic amino acids are phenylalanine. Yes, very good. Ready? Phenylalanine and tryptophan. Okay. One more is there, tyrosine. But tyrosine is actually synthesized from phenylalanine. So you can simply write phenylalanine. Isko you can remember as PAT. So PAT is phenylalanine and tryptophan. And this tryptophan has an extra imidazole ring attached to it. Okay. So tryptophan is a heterocyclic amino acid also, but यहाँ पे we are taking the standardized classification. Then coming over to third is hydroxyl group containing amino acids. Hydroxyl group. Under this will come serine and threonine. Beta, now ready, I'll just try to correct this. See, you are writing tyrosine, beta. Tyrosine is having a phenoxyl group. It has a phenol ring attached to a hydroxyl group. Okay, so hydroxyl group mein basically why we consider serine and threonine, because it has this type of group, no? And yahan pe the entire structure of amino acid is there. So tyrosine we cannot classify it under this hydroxyl group, because I'll just see, this is the structure of, say, phenylalanine. Okay, here tyrosine is there if hydroxyl group comes. So tyrosine is derived from phenylalanine. And tyrosine, since you are a FMG student, I would just like to mention this. In those patients who are suffering from phenylketonuria, phenylalanine hydroxylase is absent, tyrosine becomes an essential one, because we have to supplement them for tyrosine. Got my point ready? Okay? So this is the structure of tyrosine. Right now we are discussing only the standardized classification. Yeah, ready, you can add it also, and when we will discuss the metabolism, I'm going to discuss these structures. Right now, why I'm not discussing the structures? Because I don't want to scare first year students, because they will get, they will get a kind of this thing, acha, we have to learn this. Slowly, I will take you to that level where you will find it interesting and you would love to learn them. Okay? So I will discuss, don't worry. So this is having hydroxyl group. Then we come over to sulfur containing. Sulfur containing means they are having sulfur in them. It is clear from the word itself. So there are only two sulfur containing amino acids, cysteine and methionine. Yes, very good. Cysteine and methionine. They are having sulfur in them. Okay. Then after this, we come over to acidic amino acids. Acidic amino acids are those acids which have more acidic environment in them, and that acidic environment will come from carboxylate ion. It's clear. Okay, so acidic amino acids will have one more COOH group along with the basic ones. An acidic example is aspartate and glutamate. अब now see beta, even if you are writing aspartic acid and glutamic acid, then also it is the same. So you don't have to get confused. Yes, very good. Ready? Aspartate and glutamate. Okay. Their derivatives are also there. Asparagine and glutamine. But that will come under derived amino acids. Abhi we are only focusing on the standard ones. Clear? Then after acidic, then we come over to the basic amino acids. अब basic amino acids का मतलब क्या हुआ? Basicity is more. That means NH2 group is more. That means they have an extra amino group. And the example of this is lysine, arginine, and histidine. Basic lysine, arginine, and histidine. Clear? So I can simply say, pehle let me just quickly tell you, aliphatic amino acids are there. This is the classification as per R group. So those who are studying in first prof of MBBS, they can straightforwardly get a question in the exam that write a short note on aliphatic amino acids. Right? Aromatic, hydroxyl group, sulfur containing, acidic, basic. And then we come over to last but not the least class, which are known as amino acids. Now, what are these amino acids? Okay, they are not known as amino acids. Why they are known as amino acids? Yes, and why they come in the category of this. So, amino, first of all, the example is proline and hydroxyproline. Right. Now, first of all, beta, why they are known as amino acids? Because if I'll just, here I have drawn the structure of a generalized amino acid where you can see it has an alpha carbon atom. Okay. So, alpha carbon atom is free. In a sense, it has one amino group. It has this. Proline and hydroxyproline do not have a free alpha carbon atom. Their structure is like this. So alpha carbon atom is this. This is the alpha. So what happens is, just because of this, they are known as amino acids. But why they come under the category of amino acids? Just because of one plain and simple reason, that they resemble the properties, characteristics of the amino acids. And basically this proline is very important. It plays a very important role in the synthesis of collagen in the body. You know what is collagen? Collagen is the most abundant connective tissue which is present in the body. Yes, ready, very good. This is a FMG question. That is why you are writing correct. Proline. What is the reason behind this? Why it disrupts alpha helix? Because it does not have an alpha carbon atom. If it cannot have an alpha carbon atom, how can it react with other molecules? That is why. Yes, and collagen mein proline combines with glycine and lysine. This we will discuss, don't worry. So, but why it is known as amino acid? Because they are having, you can say bound, yeah, they do not have a free alpha carbon atom. But why they come under the category of amino acids? Because they react with amino acids to form very important compounds, and their characteristics and properties resemble to that of so many amino acids. Clear? So this is the classification as per R group. And in this R group, always remember this glycine is considered to be the simplest amino acid. Right? Glycine is considered to be the simplest amino acid. And वैसे भी it holds number 1 because I started my lecture with glycine. That's why. Clear? So this is the classification as per R. Second, we come over to as per nutritional requirement. Now, if we talk about the second way of classifying, second is as per nutritional requirement. Now, if we talk about as per this, there are 3 types of amino acids. Okay. And these 3 categories include, one is essential amino acids. Second category, semi-essential. And third category, non-essential. Yes, very good, ready? Essential, semi-essential and non-essential. How many standard amino acids are there? 20. Now these 20 will be categorized into this. So 8 are essential, 2 are semi-essential, and rest of the 10 are non-essential. Right? Now what are essential amino acids first of all? अब देखो essential amino acids are the ones essential means important, isn't it? From the definition only we can say it is important. Yeah, very good, ready. So essential amino acids, for how will we define them? We will simply define them as the amino acids which synthesized, they cannot be synthesized in the body, which cannot be synthesized in the body and are supplied from the outside diet. Right? That is why they are essential. Yes, very good, ready. Yes, they cannot be synthesized in the body. That means they have to be supplied from outside diet. Why? Because they are required for the normal functioning of the body. Because always remember, beta, just suppose most of the students who are, I mean, like the boys, in fact, girls also, I should say, you people must be gym freak. Gym. Okay, so you have that kind of thing now, when you are youngsters, you want to make your body fit. Biceps bhi hone chahiye, triceps bhi hone chahiye. They should be, proper toning of the body, the physique, I should say. So whenever you go to the gym, if you hire a trainer, the trainer will always advise you to undergo, different kind of proteins only. They will advise you to have proteins in your diet, because proteins play a very important role in building up of the muscle mass. Right? So that is what is the importance of these essential amino acids also. Up 8 are there. For this you have to remember this mnemonic. वैसे तो there are so many mnemonics, but I follow this mnemonic. TV. Yes, very good, Balaji. Yes, but how you can remember it? No, TV till 8 p.m. means you can watch TV only till 8 p.m. After that, you won't be able to watch the TV. Yes, this is the mnemonic which I use frequently. Otherwise, TV, Tim Hall, so many mnemonics are there. But this is easier to understand. TV, TILL, 8. 8 is like this, PM. So T stands for tryptophan. V stands for valine. Yeah, valine. It depends on you how you pronounce. This T stands for threonine. I stands for isoleucine. This L stands for lysine, this for leucine, this for phenylalanine, and this for methionine. So these are the essential amino acids, TV till 8 p.m. And then we come over to the second category, semi-essential. Up semi-essential means partially they are essential. Because just now I told you that the requirement, amino acids are required for the proper growth and functioning of the body. So semi-essential are the ones are required by. Okay? What if essential amino acids are not supplied? But it won't cause any mutation. Mutation though is a very big term. But yes, it will change the entire things of the body. Because देखो, अब चलो, I'll just give you one common example. ठीक है, Asif? Because if I talk about phenylalanine, suppose if I talk about phenylalanine, right? So abhi just now I told you na it is an essential amino acid. So phenylalanine, why it is essential? Because it synthesizes tyrosine in the body. Okay, ये जो tyrosine है ना, this is responsible for the synthesis of melanin. Melanin, you know, the skin pigment. जैसे we have a, जैसे you have studied in Mendel, you know, laws of inheritance. Albinos are there. Albinism is there. Isn't it, Asif? So suppose if I say phenylalanine, we don't get it from the diet. For example, tyrosine will not be synthesized. Then melanin will not be synthesized. Isn't it? If melanin is not synthesized, that characteristic color to the skin will not be given. And the person will suffer from albinism. मतलब simplest language में I am giving you the example, Asif. Clear? So what I'm trying to tell you here is, if essential amino acids are not supplied, they won't lead to any mutation, but yes, so many important derivatives will not be synthesized in the body, which are required by the body actually. Delayed growth will be there. You can say the person can be mentally retarded also. So many things. Clear? Yes, in a nutshell, yes, I can explain it. It can explain the entire metabolism of the body. Asif, clear, beta? Same is the case with methionine. Methionine said so many other diseases are caused. Leucine also so many diseases are caused. So that is why they are essential. Ab don't think like that, Asif, ki non-essential are not important. Non-essential are also important, but they can be synthesized in the body easily. Right? When you will study the metabolism, beta, it will be clear to you. Now, semi-essential are required. Halki you are a NEET UG student, so metabolism won't be of much help to you. But yes, this lecture which I am taking today will be very beneficial for you. So, semi-essential are required by growing children, first of all. and lactating mothers in more quantity. That is why they are semi-essential. Okay, and for them the example is, there are only two. Just now I told you, arginine and histidine. Yes, arginine and histidine. Very good, Balaji. Yes, arginine and histidine are the semi-essential amino acids. Now 8 plus 2 10, 20 minus 10 10. Rest are non-essential. Rest are non-essential. Now, be it glycine, so many are there. Be it cysteine, they are all non-essential. They are required by the body, but they can be easily synthesized, so the body does not have to worry. Body does not have to take them from outside. Okay? Wow, very good. Then so you will become a master of biochemistry. Yes, right. Okay. And alanine, asparagine. Yes, yes, Balaji, you are right. They all are non-essential. But I just want to tell you one thing, that asparagine and glutamine, they are derived amino acids. Okay, they won't come under these. These are only standard amino acids. They will come under derived one. So rest of the one you are correct, Balaji. They will come under non-essential. And Asif, this will be very beneficial for you, because once you go into MBBS, these are the lectures which will actually help you. Okay. So this is classification as per their nutritional requirement. Then third way of classifying is as per their metabolic rate. Right. Now, if we talk as per their metabolic fate, this is the generalized structure. Again, I will just write it. Abhi, just now we have studied that there are 20 standard ones, and depending upon the R group, they are different. That means in all the 20 standard amino acids, this structure will remain the same. Obviously, the structure which will vary, that will be only the R ones. Okay, so now how we classify them? Either they will be having a different metabolism as per nitrogen, or they will have a different metabolism as per carbon. Okay, so how we say as per the metabolic fate? As per okay, beta, because you know this, you are right. In some books, essential are said to be 9 involving histidine. What can you tell? Because beta, sometimes what happens is this histidine cannot be synthesized in adults. That's why. Getting my point? Matlab, what happens is the ideal, you can say, classification is essential, semi-essential. Okay, this can be synthesized in adults also. That's why it won't come under that category. So it will be placed separately. If you see standardized biochemistry books, their semi-essential will be there. Yeah, if you see in nutritional biochemistry, this will be the one which is covered separately. And if you see the book correctly, okay, you just check what is the difference between histidine and arginine there. So there they don't consider this arginine. And वैसे भी this histidine is considered to be essential because it is required. Why? Because it is the most, I should say, uh, stable amino acid at the intracellular pH. Because it is having, I'll show you the structure also, it is having that special ring in it. Okay, so histidine, A slight change in the pH will change the entire concept, yeah, entire structure of this. So histidine is considered to be the most stable amino acid at the intracellular pH. So essential you can say 9, but ideally, that is why beta, yes ready, that is why it is required for the synthesis of buffers. Just because of this reason. Because histidine is a very stable, it is a very effective amino acid. And one more example of this, histidine is present in the structure of hemoglobin also. Hemoglobin, where there is a phytol tail, histidine is present. Right. And just because of this reason. And that is why it is placed under essential ones also. But overall, IOR, it is a semi-essential one because histidine ki requirement, it increases in pregnant mothers basically, and in lactating mothers also it increases, and in growing children also it increases. Is it clear, IOR? And just now Reddy mentioned why it is an example of physiological buffer, because it is the most stable amino acid. Slight change will be there. So like it is more stable there. Clear ho gaya beta? Sorry, IOR, is it clear to you? If not, I'll explain again. Right? Then we come over to the last one. Yes, as per the nitrogen skeleton. Now, if we talk about nitrogen skeleton, okay, is it clear, IOR? IR? Right. Okay. So if we talk about as per nitrogen skeleton, you know, proteins or amino acids are the only molecules in the body which have this nitrogen in them. You saw the structure of carbohydrates? Yeah, मुझे भी यही लग रहा है. Okay, so as per nitrogen skeleton, so proteins are the only macromolecules which have this nitrogen in them. And you know this nitrogen which is present in the form of, okay, clear. Okay. You are from which place, Iyer? Are you from Southeast? So, you know, since amino group is there, nitrogen is there, this is amino group, you know how it is there in the body? It is present in the form of ammonia in the body. And this ammonia, it comes out from the body in the form of urea. Urea, which is a very important, I should say, just we are ureotelic. Why we are ureotelic? Because the excreta is urea in us. And you know what is the major source of urea? Urea is coming from the proteins. And from where? From this amino. So, as per nitrogen skeleton, this urea needs to be removed from the body. Otherwise, what will happen is, just say if I take your example, suppose if you are constipated, say if you don't go for motions for approximately 4 or 5 days, you will start feeling a kind of, you know, discomfort will be there. Isn't it? You will try all laxatives so that you can go to the restrooms and you can just relax. So this urea plays a very important role. It actually disturbs whatever is we are thinking, means it disturbs the normal functioning of the brain. So we have to remove this urea. And how it is removed? It is removed by three processes: transamination. Right. Abhi, right now just stick to this. Transamination means first of all amino group is transferred. Then this amino group is removed, deamination, and then it will form urea. So urea cycle. So this is metabolism as per nitrogen skeleton. Yes, very good. Trans deamination beta, that is for glutamate. That also I will explain, don't worry. Okay, abhi right now though they are just trying to understand the basics na. So abhi just stick to transamination, deamination, and then urea cycle. Okay. Transdeamination is a coupled process only for glutamic acid. Then second is as per carbon skeleton, isn't it? So second will be as per carbon skeleton. Now, if we talk about as per carbon skeleton, they are categorized into 3 subcategories. This is very, very important. 1 second. Acha, here you have written in okay, South Sudan University. Now I got it. Yeah. So if you have any problem, you can, this is my number. I'll just write my number here. You can connect to me on this number also. If you have any problem related to, like the topics which you have not studied with me, so you can just message on this number. And coming over to your query, trans deamination bhi hota hai beta. Ready? It is there, but it is for glutamic acid. Here, yes, we have written separately. Okay. So this is my number. Now, so as per carbon skeleton, these amino acids have 3 different modes. One is glucogenic, right? Second is ketogenic. Okay, I'll explain these terms separately. And third is glucogenic plus ketogenic. Yes, very good. So glucogenic, ketogenic, and glucogenic plus ketogenic both. So this is the fate as per carbon skeleton. Now obviously you will be anxious to know what are glucogenic amino acids. First of all, you have to remember these 3, then automatically glucogenic amino acids will be clear. So out of these 20 standard amino acids, 2 are ketogenic. Okay. For this, you have to remember the mnemonic as LL, which is leucine and lysine. Right. Then coming over to both glucogenic and ketogenic, it is PITT, PIT. Okay. So you can say 4 are both. Now 4 plus 2 is 6. So 20 minus 6 is 14. So 14 will be, the rest of them will be glucogenic in action. Right. Now if we talk about the both, P stands for phenylalanine. I stands for isoleucine. T stands for tyrosine. And this T will stand for tryptophan. Yes, very good. Yes, glucogenic, yes, very good. Biology, very good, ready, yes. Right. Okay. Rest of them are glucogenic. Now coming over to your FMG question, I'll just say glucogenic में the most glucogenic amino acid is alanine. Alanine is the most important glucogenic amino acid. Right. So this is the classification. Yes. बाकी जितने भी हैं all will be, rest are glucogenic. right? Just now, be it alanine, be it valine, be it glycine, कुछ भी होगा, anything apart from 4 plus 2, 6, rest of them will be glucogenic. So this is your classification as per their C group or as per their N group. Till here it is clear to you, beta? Right? Now the last classification which is left here is as per their charges. So as per their charges, we will be discussing in the metabolism. The reason I'll tell you why, because overall I can simply say as per the charges. As per the charges, we can classify them into polar and non-polar. We can classify them as charged and we can classify them as uncharged. Right. So one example I'll just quote here just for your reference. Yeah, just for your reference, because we are going to discuss them once I write the structure. So I'm thinking I'll write the structure along with metabolism, because right now I don't want want you people to expose with the structures. Slowly we'll move. Okay. So polar के लिए, for example, if I say polar, affinity towards water, simple concept है. Okay, so we can say polar charge में aspartate will come, obviously, glutamate will come, serine will come, threonine will come, like that. One example I am quoting, lysine arginine will come. And if we talk about non-polar जैसे all the aromatic amino acids will be non-polar, जैसे phenylalanine is there, right? Tryptophan is there, it will come under non-polar, like that. So this we will be discussing when we will discuss the structure. So this is the classification of the amino acids as per the nutritional requirement. If I talk about as per nutritional requirement, positively charged or basic, yes, they will be charged only. Yes, you're right, Reddy. That's why I've written a lysine, arginine are there, serine, threonine will be polar, aspartate, glutamate will be charged, like that. So once I write the structure, then it will be more clear to you. And for you people, Reddy, Balaji, for you, understanding metabolism is very important, because the questions have been asked mainly on those topics only. Okay. So as per nutritional requirement, I can say that amino acids are, you can say, essential, semi-essential, non-essential. As per their metabolic fate, they are your this thing, glucogenic, ketogenic, and both. And as per nitrogen skeleton, transamination leading to deamination, leading to urea cycle. Right. Now, after this, then comes how these amino acids are going to form proteins, because here we are focusing on the proteins. So I can say that question is how proteins are formed. Right. If we talk about how these proteins are formed, I can simply say that one amino acid will combine with the second amino acid. Clear? So when one amino acid combines with the second amino acid, it loses a molecule of water. So obviously when it will lose a molecule of water, both the molecules are connected, a bond will be produced, and this bond will be known as peptide bond. And you will be surprised to know that peptide bond is the strongest bond. Even if the protein is denatured, yes, very good, ready. Even if the protein is denatured, kuch bhi kar lo, whatever we do with the protein, we heat it, we put an acid, we put an alkali, anything we do, the peptide bond will always remain intact. Okay, so this is the only bond which can be only broken with the help of sequencing methods, not with any of the you can say external method like acid, alkali and all. Right? Now see what happens is, say this is one amino acid, NH2, COOH, H and R. Right? This is second amino acid, COOH, NH2, H and R. Okay, I can simply say water molecule will be given out like this. That means minus water. So we get this type of structure. CH, NH, C double bond O, then C, NH2, H, R, COOH and R, isn't it? Now what is this? This is your peptide. 4 atoms scope planar linkage. Yeah, very good. Then ready, you must be knowing about Ramachandran angles also. What are Ramachandran angles? Very good. You know, we have to write it like this basically, NH, C double bond O. Okay. So this is your, I will just erase it, otherwise you'll get confused. Okay. So this is what we call it as a dipeptide. Right. If we put third amino acid, it will be known as a tripeptide. Yes, very good, Asif. Ramachandran angles, basically, Ramachandran was the scientist who said like ये ऐसे होता है ना, C double bond O NH. So he said there are two type of angles, psi and phi. Okay, so for a fully stretched peptide, it will be linear like that. But this is again, you people study in class 11th and 12th. Baki usme it is never asked. But yes, Ramachandran was the name. Ramachandran angles. ठीक है? Right. So yes, ready, yes. Psi and phi, yes. Between C and N, basically the reactions is between C and N, no? That's why. And C and C. Okay? So this is a tripeptide. So dipeptide, then tripeptide, then it will become a polypeptide. Okay? So if I say two amino acids combine, if I say, second, two amino acids are combining, it is a dipeptide. If 3 amino acids, it is a tripeptide. And if more than 3, it becomes a polypeptide. Right? Now, if I talk about a polypeptide, I can say that they will all have two type of terminals. एक हो जाएगा C terminal और एक हो जाएगा N terminal like this. Partial double bond character, that is why they are resonating also. That is why beta this is the strongest bond. Yes. Suppose, वैसे तो क्वेश्चन नहीं आता है, but suppose if somebody asks you कि why it is strong, it's the strongest bond, the reason is it is the strongest bond because of the resonance in them. Right. Right. So it has a carboxyl terminal and it has a N terminal. Always remember. So this is how they are made. Clear? Now, if I talk about, this is how these, how proteins are formed. So you can say proteins are formed when one molecule of amino acid combines with another amino acid, losing a molecule of water to form a dipeptide, tripeptide, and polypeptide and so on. Now the question is, what are the biologically important peptides? Biologically important peptides. Right? Now, if I talk about a dipeptide, okay, the example of dipeptide is aspartame. This aspartame is formed of two amino acids. One is your phenylalanine and second is aspartate or aspartic acid. Right. So I can say that this aspartame is produced when phenylalanine combines with aspartic acid, it loses a molecule of water. What is its significance? The significance is it is an artificial sweetener. Artificial sweetener. Right? And what is the significance of this? Just say if we talk about sugar free tablets, you know that yellow color this thing comes, so instead of putting sugar, this is put. So basically it is used for sweeteners in so many different, whether it is candies, basically it is sugar free also. Okay, so this is the significance. Then if we combine three amino acids, then what it is going to form? It is going to form a tripeptide. And if we talk about the example of tripeptides, there are two very, very important tripeptides. One is your glutathione, right? And second is your creatine. You must have heard the name of both glutathione as well as creatine. Isn't it? So if I talk about glutathione, we all know that this is, since this is a tripeptide, that means it is formed of three amino acids. Thanks, ready for giving the heart. That means you people are understanding. I am happy to know that. Thank you so much for understanding it. But beta, my sincere advice, again I'll tell you one thing, that try to attend the classes on a daily basis, because see, my time is fixed for the class. The day I started my classes, I have kept the timing just because of you people, because so that you also become, a habitual of attending the class at 11 o'clock till 12:30 max 1. Okay, so automatically you will be conditioned to study. Obviously, you have to put in your efforts, because right now most of the students are having their vacations. You people become lazy to get up in the morning. But even I am also getting up early morning to get ready to take your class. Okay, so initial efforts you make, and everything will be, easier for you people, because this is the right time when you can devote maximum number of hours on your concepts. ठीक है, so glutathione के लिए you have to remember this GCG. To remember, GCG stands for glycine, C stands for cysteine, and G stands for glutamate. Okay, now what will be its function? We know cysteine is having SH group, so this is the most important antioxidant. Glutathione, antioxidant. And there are so many questions. If I talk about FMG only, the yes 11 a.m. is the fixed time for my class. It will be from 12 till 12:30 or 1, because I know it is difficult to study at a stretch for 2 hours. So I have made it till the time the topic is finished, and I try to finish my topic by 1.5 hours, so that I don't want to put extra burden on your brains also. Okay. So glutathione is an antioxidant. A bit of antioxidant, it is clear from the word, which is reversing oxidation. So oxidation is reversed in the body, it is stopped in the body. That is what is its use. And coming over to this ready, yes, glutathione reductase and glutathione peroxidase. These are the two very important enzymes. Glutathione peroxidase requires selenium and glutathione reductase requires B2 or riboflavin, which is again a very important question from examination point of view. Okay, so glutathione, what is it? It is formed of GCG, glycine, cysteine and glutamate, and abhi right now I'm just writing it is an antioxidant. Slowly you will come across the functions when we will discuss glycine. Okay, then second is your creatine. What is creatine? We all know that creatine is the energy reservoir of the muscles. Energy reservoir in the muscles. Right? And we all know that creatine INA. Okay. The mnemonic is JAM. Halki the spelling of JAM is JAM. Here you can just remember GAM you can say. Okay. So GAM means it has glycine, arginine and methionine. So glycine, arginine and methionine forms creatine in the body, which is the energy reservoir in the muscles. Whenever the muscles are contracting, energy is coming from creatine. And you know this creatine, this is a very important reaction also. This creatine forms creatine phosphate. And the name of the enzyme here is CPK. Ready must be knowing the importance of CPK, isn't it? And ATP is required. In myocardial infarction, CPK rises. Okay, and you know this creatine, when it gives out a molecule of water, it forms creatinine. Creatinine. What is creatinine? Creatinine is an anhydride of creatine. Anhydride you can very well understand. Anhydride of creatine. And what is its function? Creatinine clearance test. Yes, very good, ready. Creatine phosphokinase, yes, CPK, and that too CPK2MB is the fraction which rises in MI. Okay, so creatine will be converted into this anhydride of creatine and creatinine clearance test. It is basically done to check whether our kidneys are working fine or not. It is known as creatinine clearance test. So this is what is a tripeptide. And thus, if we talk about polypeptides, so they are many. You talk about all the peptide hormones. Peptide hormones will come under this category. Right? You start from insulin to glucagon to oxytocin. So many are there. Oxytocin, vasopressin. They all are peptide hormones. Right? So this is what is the significance of the amino acids and how these amino acids are being converted into the proteins. Is it clear? I hope till here it is clear to all of you. The most important which point about which you should actually know is about the PI, isoelectric point, because isoelectric point is the point which is actually responsible for differentiating the amino acids from proteins, the basis of separation. Right, okay. Okay. So this is, I think, all about the properties. Now, one more thing, if I have to tell you, I'll just write it. Properties of the amino acids, okay, as per their carboxylic group and as per their amino group. isn't it? Because they are having both. So what are the properties of amino acids as per their COOH group or as per their amino group? So I can simply, just summarize everything. I can say that all the amino acids, they undergo decarboxylation. Decarboxylation means removal of carbon dioxide. So decarboxylation of the amino acids leads to the formation of biogenic amines. Right? And these biogenic amines include, जैसे if I talk about histidine, we all know histidine is a basic amino acid. This histidine will be converted into histamine, which is a very important, you can say, the substance which is released during hypersensitivity, and it is released by the liver. Okay, then tryptophan forms serotonin. Serotonin, beta, most of the students, you must not have heard this. Okay, no problem, ready. I'm also just winding up the class in another 5-10 minutes. This is the main disadvantage of this. Yes, but what you can do is, if you want any kind of help, I have given my number also. My Telegram channel is also there, so you people can have an access of the notes from there also. Okay. Yeah, or is it clear? Good. Okay, so tryptophan forms serotonin. Beta, serotonin क्या करता है? It stimulates the cerebral activity. This is sometimes what happens is we are sitting very low, and sometimes we see something. Yeah, to come out from depression, it is like it elevates your mood. It enhances your mood. So that is what is serotonin, which is synthesized from tryptophan. Okay, so these are the two I'm just writing just to make you clear. So these are the two biogenic amines which are formed as a result of decarboxylation. There are so many. Jैसे tyrosine forms tyramine, like that. Then second important one is ninhydrin test. Right? Beta, you can join my Telegram group. It is Biochemistry by Dr. Pooja. Otherwise, I have shared my phone number with you, so you can just drop a hi so that I will share my Telegram link with you there, so you can join it. So it's up to you. Okay. Then comes ninhydrin test. Ninhydrin test is basically again decarboxylation. And what happens is, this is a confirmatory test for the proteins or the amino acids. Right? And proline with ninhydrin test, it will give yellow color, and other amino acids will give a Roheman purple color. Purple. Right? So these are two of the important properties. Decarboxylation, where you can simply say amino acids undergo decarboxylation to form the biogenic amines, and ninhydrin test. Ninhydrin test is a very important confirmatory test to check the presence of the amino acids in that. And rest of the properties will be simple, because amino group is there, HCl is there, so you can say COH is there, so they can combine to form salts. So many are there, right? So I think we can conclude our lecture till here. I hope it is clear to all of you. Till here, if there is any confusion, you can ask me. So I'll just quickly summarize what all I did. Basically, what all we discussed today. We discussed basically the properties of these amino acids, right? I told you about their R group classification. I told you about their metabolic fate. right? Then I told you about the most important one, isoelectric point, right? Then I told you about the as per the nutritional requirement. And the most important thing I told you about the peptide bond, which is considered to be the strongest bond, because in the next session we will be discussing about the characteristics and properties of the proteins. So if you know they are formed from the amino acids, then you can clearly correlate them with the proteins. And then we studied little bit about the characteristics of this. All are very, very important because these are the basics on which the entire, you can say, chemistry of proteins is also based. And yes, we discussed this also, biologically important peptides. So I hope it is clear to all of you. Is it clear, beta? And if you guys want to join my Telegram group, this is my Telegram. This one. So you can join the Telegram group also. Asif, you can contact me on this number. We have a solution to every problem, don't worry. Okay? And you got very good marks, in fact, I should say. So connect to me on this number. So today we are going to wind up here. So if you guys don't have any problem, then we can wind up the session. And in the next session, because in the next session we will be discussing about the properties of proteins, and then we will move towards the metabolism. Clear? So I hope everything is clear. Yes, you can connect to me on this, then I'll guide you. Right. So it's clear to all of you? Yeah. Okay, my pleasure. So see you guys in the next session. My session always starts in the morning since tomorrow is Sunday. So Sunday there won't be any class. That's a leisure day for you people and for me also. So then we will connect on day after. Okay. So wish you guys a very good day and a very good weekend. So see you guys later. Till then, bye bye. Take care. Yeah. Bye.